Fluorescent microscope. What is the definition of fluorescence microscope? It's an optical microscope that uses fluorescence and phosphorescence to study the properties of organic and inorganic substance. We'll see later what is fluorescence and phosphorescence. So it was devised by August Kohler, Carl Richard, and Heinrich Lehmann. There are two types of fluorescent microscopy. So one is called epifluorescent microscope, which we are going to discuss in this topic. And the second one is the simplest one. Epifluorescent microscope is the simplest type of fluorescent microscope. And the second type is called confocal microscope, which uses the optical sectioning to get a better resolution of the fluorescent image. This is a fluorescent microscope, and here you will get the image. So this is a fluorescent image. So let's discuss some of the basic concept of fluorescent microscope. So the light emitted by an energized molecule is known as photoluminescence. If this is a substance, if it emits some light, if this is energized, if this molecule is energized and it emits light, is called photoluminescence. So if this is the energized molecule, if it emit light, then the process is called photoluminescence. So in this process, there is a time lapse between the absorption and emission of light. For example, take here. So this is the specimen. This is the object. And this is the light. So this light is absorbed by the specimen. And later, after a fraction of a second, it emits a light of different wavelength. So it absorbs the light and it emits a light. So there is a time lapse between the absorption and the emission of light. So based on that, there is phosphorescence and fluorescence. There are two types of based on this. If the time lag is greater than 1 by 10,000 of a second, then this process is called phosphorescence. And the fluorescence is if the time lag is less than 1 by 10,000 of a second, then the process is called fluorescence. So there are phosphorescence and fluorescence. The substance that emits fluorescence is called a fluorophore or fluorochrome dyes. For example, there are green fluoroproteins, red fluoroprotein. So which emits green light, here it emits red light. So that is the difference. So the material can be made fluorescent by combining with the different dyes. So this is a basic concept about the photoluminescence. Now let's discuss the outline of what is a phosphorescent microscope. So there are 10 components. So the 10 steps are there. First one is the light source, which emit light, and the second is the excitation filter, which absorb almost all type of colors, but it pass only one type of light. Here it is the blue color. So this blue light will be reflected from the dichroic mirror. So this is the dichroic mirror. Then it passes through the objective lens and get absorbed by the specimen. Here the specimen is stained with a fluorescent dye. So after the absorption of this light, blue light, it emits another type of light, which is green in color here. So this green color wave light passes through the objective lens and it passes through the dichroic mirror. So in the first case, it reflect the blue light, and in the second case it allow to pass through it. That is the significance of dichroic mirror. It allows transmission and also reflections. So dual functions are there. Then in the eighth one is the emission filter, which blocks certain unwanted or harmful rays. Then the fluorescence light pass through the ocular lens and get viewed through the detector. So this is a outline of phosphorescence microscope. Now here we already discussed a source of light. So which this light will be absorbed by the fluorophore which is present in the specimen. So suppose this is a specimen. If this absorb only one particular type of light, say blue color. So the blue light is absorbed by this specimen. When it absorbs the blue light, the electron in the fluorophore molecules in the dyes are get excited. So they move to a higher energy state. Here it is ground state. The electrons are in the ground state. When the photon strikes the fluorophore molecules, the electrons in the fluorophore molecule get excited and it goes to a higher energy state. And it for a short fraction of, or a short time or a fraction of second, and then come back to again into the ground state. So while coming back to the ground state, it release or it emit light. It emit light of higher wavelength. If it is blue light, if it absorbs the blue light, then it emit another wavelength or higher wavelength, a slightly higher wavelength. Here it is a green light. It will emit the green light. So this emitted light can be viewed through the detector, can be shown through the detector. So this is the basics of fluorescent microscope. So this is a much more detail, and this is the fluorescent molecule. So this is the representation, and nucleus, protons and neutrons are there, electrons are in the orbit. So this is the excitation light. So this one is this blue light is excitation light. So the electron absorbs these photons and get excited and move to higher energy level. So this one. So this is here is it moves to the excited state, and this is only for a fraction of a second, is very unstable and come back to the ground state. So while coming back to the ground state, it emits a light which is of different wavelength, and this process is called a fluorescence or emissions. So when an electron from a higher energy state moving back to the ground state, it emits a particular type of light, and the process is called a fluorescence. So yeast cell membrane visualized by some membrane protein fused with a red fluorophore and green fluorophore. This is what the pictures look like in fluorescent microscope. And what is the principle of fluorescent microscope? The basis of fluorescent microscopy is to stain the components with the dyes. A special type fluorescent marker is used here. The molecule absorbs excitation light. Here the fluorescent molecule absorbs the light, excitation light at a given wavelength, maybe blue or something like that, given wavelength, and after a short delay it emits light. It absorbs the light, move to a higher energy state, and then emit light. So this is the principle of fluorescent microscope. Longer wavelength. So before excitation, the electrons are in the lowest energy state. It's called S0. Lowest lowest energy state available to the ground state is called S0 state. So when it absorbs the energy, it moves to the, depending on the energy level, it moves to the next orbit, S1, S2, S3, S4 or S5, different state. And then it comes back to the ground state and it emits light. So here it absorbs the blue light and emits the green light. So while coming back to the ground state, it emits a longer wavelength and with lesser energy. So when an electron hit with a photon of certain energy range, the molecule absorbs the energy of the photon and jumps up to a higher energy state, which is S1, S2 or S3. This is why emission of light has a longer wavelength. So it has a longer wavelength than the excitation light. This one. അപ്പോൾ ഇതിനേക്കാളും കൂടുതൽ wavelength ഉള്ളതാണിത്. But here the energy is less. Here the energy is much more greater. And they differ in the color. So this is the blue color, this is blue color, and this is green in color. So what are the steps involved in fluorescent microscopy? First, uses a higher intensity light to eliminate the sample. So the first steps in the fluorescent microscopy is use of high intensity light to eliminate the sample. So this is the light source. The light excites the fluorescence molecule which is here in the sample, which then emit a longer wavelength. The emission light can be then viewed through the detector to reveal the location of fluorophore. So what are the components and working of a fluorescent microscope? There should be a fluorescent dye. So this is also called a fluorophore. It's a marker. So a fluorophore is a fluorescent chemical compound that can re-emit light upon light excitation. Many fluorescent stains have been designed for different biological molecules. Some of the examples are the nucleic acid can be stained through DAPI and hotchest and DAPI. These are the two fluorescent markers which can be used to stain the nucleic acid. Phalloidin is used to stain the actin fibers in the mammalian cells. So these are the images of the these are the fluorescent image obtained through the fluorescent microscope. So what are the components of the fluorescent microscope? First one is the light source. So this is the light source. So the four main types of light source are used, including xenon arc lamps, mercury vapor lamps, lasers, and high power LEDs are used as a light source. Second is the excitation filter. This is the excitation filter. So it's typically a band pass filter that absorbs the light. It absorbs the light and passes only certain wavelength of light specific to the fluorophore. So this it allows only a particular type of light which is specific to the fluorescent dyes used in the specimen. Here it is the blue light. It passes here. The excitation filter absorbs all other light but only pass the blue light here. Then comes the dichroic mirror. This one, dichroic mirror. So it's a thin film filter. It's a very accurate color filter used to selectively pass light of a small range of colors while reflecting the other color. So here it reflects the blue colors and transmit the green color. So that is the significance of dichroic mirror. Next one is the emission filter. This is the emission filter, which is typically a band pass filter that passes only a wavelength that is emitted by the fluorophore. ഇവിടുന്ന് ഈ എമിറ്റ് ചെയ്ത ലൈറ്റിനെ മാത്രമേ അത് പാസ് ചെയ്യിക്കത്തുള്ളൂ. So this emission filter allows only the emitted light to pass through it and block all undesired light outside this band, especially the excitation light. So it blocks the unwanted excitation light, including ultraviolet and infrared rays from the samples, and optical filters ensures the darkest background. So this the emission filter ensures the background is darkest in conditions. And next one is the ocular lens. This is a normal function, helps to amplifies the image. And finally, these images can be viewed through a screen, and this is called the detector. So this is all about the fluorescent microscope. And what are the applications of fluorescent microscope? It helps to identify structures in fixed and live biological samples. It is used in multicolor staining, labeling of structures within the cells, and measurement of a physiological state of a cell. What are the advantages of fluorescent microscope? Is the most popular method for studying dynamic behavior exhibited by the living cell imaging. This stems from its ability to isolate individual proteins with a high degree of specificity amidst non-fluorescent material. That is used to isolate a particular type of specific protein. Different molecules can now be stained with the different colors, allowing multiple types of molecule to be tracked simultaneously. So each molecule present in the cell can be uh, stained with the different colors, green, red, or something like that. Here it is yellow color. You can see the blue light color, green light other. So the different organ can be, or the different material can be stained with the different colors. And the last one, these factors combine to give fluorescent microscopy a clear advantage over the other optical imaging technique for both in vitro and in vivo imaging. That's all about the fluorescent microscope.